Okay, this is an overview of my presentation, and not surprisingly, there are two main parts. I will talk about 10 to 15 minutes about genotoxicity and the remaining time about carcinogenicity. But first of all, I would start with a brief discussion on what is relating to the title of this session this afternoon. Overcoming specific non-clinical challenges, and I will discuss why these three toxicological endpoints cover in this session: genotoxicity, carcinogenicity, and also reproductive toxicity, why they are so special and different from other toxicological endpoints, and why may there be specific challenges. And I would like to explain one of the key differences with this slide here. If you consider the relative importance of the study data, both non-clinical and clinical study data for safety assessment, for safety human assessment during the whole drug developing process, I think it's quite clear that the relevance of the non-clinical data is highest when there is no clinical experience or no clinical data, which means before first-in-man studies, or in order to assess the human safety in the first clinical trials. With increasing the clinical or the human experience and data, the non-clinical studies or data will obviously lose their relevance, and later on at the time point of marketing authorization, human safety is mainly based on clinical data. Well, and that's exactly the difference for the endpoints we are discussing this afternoon. This is not applicable, this rule, to genotoxicity, carcinogenicity, and reproductive toxicity. So even at the time when a drug is entering the market, the human safety related to these endpoints mainly depend on or are based on non-clinical data. And this is, of course, a very special situation then. Another critical point, I would say, is referring to carcinogenicity and the timing of the carcinogenicity studies during drug development. Here it is shown in comparison to genotoxicity. For genotoxicity, it's not a problem at all. I think there is, from very, very early on, a very good understanding of the genotoxic potential through efficient QSAR in silico tools and also high throughput screening tools. And the GLP in vitro studies for genotoxicities are usually done before first-in-man studies. For carcinogenicity, this is completely different. It's a very late non-clinical study. And even if you start after phase 2, it will take another 3 years for the final study report. And if we then have unexpected findings, critical unexpected findings, that could make huge troubles and may delay the developing process of a drug. In such situation, there is sometimes even the need to do additional genotoxicity studies. In those cases where there is no clear non-genotoxic mode of action for the tumor findings, where there are an indication for a genotoxic mode of action. That would be, of course, the worst case at that late development process to come up with such problems. So having this in mind, I will then talk about genotoxicity, first the guideline requirements, and then mainly about study outcome. What is the impact of certain study outcome for the drug developing? And finally, about the relationship between genotoxicity and carcinogenicity. Now, for genotoxicity assessment, it's very important to understand that different types of mutations exist. And these different types of mutations can be nicely linked to the different levels of organization of the genetic material, as shown in this picture here. So it starts with what we call gene mutation. These are changes in the sequence of the bases at the level of the DNA molecule. Then we know structural alterations at the level of the chromosome, also known as chromosome aberration. Compounds inducing such effects are known as cluster genes. And finally, at the level of the whole genome, we know numerical changes of chromosomes. These can be gain or loss of single chromosomes, known as aneuploidies, or the whole chromosome set may be changed, known as polyploidy. The latter is not that relevant for toxicological, is not such a relevant toxicological endpoint. Compounds inducing aneuploidies are known as aneupgenes. So actually, genotoxicity is looking for mutagens, cluster genes, aneupgenes. And all types can be induced by chemicals, including pharmaceuticals. All types are involved in cancer development, and there is no single genotoxicity study or test that can detect all types of mutation. That is why the guideline is recommending a testing battery. The relevant guideline for genotoxicity, as probably most of you know, is the ICHS-2. It has been revised in 2012. This guideline defines the standard battery and the design of the studies, and is also giving a recommendation on data interpretation and follow-up testing of positives, and is focusing on small molecules. And then we have heard already in the morning there are a number of different guidances that also address genotoxicity issues. We have heard about ICHS-6, S9, the oncology guideline is more or less stating that genotoxicity studies is not needed during drug development, but for marketing authorization. M3 is providing information on the timing of these studies. And a quite recent new guideline, M7, is about assessment and control of mutagenic impurities. But in my presentation, I will focus on S2 and the genotoxicity assessment of small molecules. The S2 guideline is the testing battery recommended by ICHS-2. Is mainly comprising of two in vitro studies. First, a gene mutation study in bacteria, the so-called AIMS test, and then an in vitro test in mammalian cells for chromosomal damage. There are three different options you can choose among. My advice would be to do the in vitro micronucleus test. The combination of AIMS test and micronucleus test is the best to cover the three endpoints I just mentioned on the first slides. And then this is the battery also includes an in vivo study for chromosomal damage. This is usually a micronucleus test. This can be done as an acute standalone test, but the guideline, the revised guideline, is now encouraging to integrate it into a repeat dose toxicity study. There is an option 2, which is rarely done, almost never. In option 2, there is only one in vitro study, that's the AIMS test, and the second in vitro study is replaced by a second in vivo study, which is a COMET assay in the liver. This option is not necessarily asking for an additional animal study, because the micronucleus test and the COMET assay can be easily combined in one study. The rationale for this option 2 is, if in option 1, one of the mammalian cell tests is positive, the recommended follow-up test would be the in vivo COMET assay in the liver. So this option 2 assumes that if the mammalian cell test would have been done, it could have been positive. So it's actually directly the follow-up strategy. Yeah. If combined these both endpoints into one study, then it might be not feasible to do integrated and repeat dose toxicity study. Usually this combined study is then an acute study. Now let's talk about test results. Well, the preferred situation is, of course, you do the battery and everything is negative. And in this situation, the guideline clearly states that would provide sufficient assurance of absence of genotoxicity, and further testing would normally not be necessary. This little word normally already indicates there may be exceptions, and actually 3 scenarios are mentioned in the guideline where even if the standard battery is negative, you might do more. The first scenario is that there is a human metabolite not present in the preclinical models. I think that's quite obvious that you would need to do a genotoxicity then with a metabolite. Second scenario is you have carcinogenicity findings with no clear non-mutagenic mode of action, and where mutagenic mode of action cannot be excluded. In this situation, organ-specific in vivo studies are usually recommended. And the third scenario is there is a structural alert or knowledge of class-specific effects and indicating that the negative finding is based on an inappropriate test strategy. In that case, let's assume you have a compound which is structurally related to a known anugene, and the studies were negative, and the mammalian cell test is done with a mouse lymphoma assay, which is not really the most appropriate test for anugenes. Regulators might request an in vitro micronucleus test in such a situation. Okay, more critical is, of course, positive outcomes of the genotoxicity studies, but it very much depends on what test model is positive. So the most frequent situation is a positive in vitro mammalian cell test. And as I said, in this situation, the follow-up test would be a second in vivo study, COMET assay in liver. We have done at the BfArM some years ago a review of nearly 600 compounds with regard to the in vitro mammalian cell tests submitted to us. And the outcome of this review was that 30% of all compounds were positive in at least one in vitro mammalian cell test. And that's, of course, a very high incidence. It's actually an unacceptable incidence. And most of the, nearly all were, after follow-up testing, considered to be irrelevant positives. This review was one of the reasons why we have revised the guideline in 2012. And we have included some recommendation to avoid this situation of false positives with the in vitro mammalian cell test. And here is a list of advices how to avoid the positive outcome, which is considered to be not relevant. The first is that with the revision of the guideline, the top concentration has been reduced from 10 to 1 millimolar. Then it's recommended to do appropriate measure of cytotoxicity, which are defined in updated OECD guidelines, and not exceed the limit of cytotoxicity as indicated for each test model, because high cytotoxicity is very often related to irrelevant carcinogenicity effects. Not to test into the precipitating range. The old guideline S2 was recommending exactly that, and this has led to sometimes to false positives. And finally, the use of appropriate target cells, and this is mainly target cells which are proficient for the tumor suppressor gene P53. So some of the very widely used in vitro mammalian cell types, mouse lymphoma, Chinese hamster cell line CHO, CHL, V79, are P53 deficient, and that's the reason why they are prone to false positive. So the recommendation is to use human lymphocytes or human lymphoblastoid cell lines. A different situation compared to positive in vitro mammalian cell tests, which can be easily handled, is a positive Ames test. That's a rare event, and usually it would trigger termination of drug development. Actually, we as regulators don't see any positive Ames test because, well, the development is usually stopped. And they are very efficient in silico tools very early in drug development to filter out such candidates which would be positive. There might be cases where a metabolite is discovered which is Ames positive. And if this occurs late in development, that can bring you really in troubles. Usually in this situation, in vivo test is done, which is usually not done, but is appropriate to follow up in vitro mutagenic compounds, and that's the transgenic gene mutation study. The same situation for the in vivo test. If these are positive, which is also quite rare, that would be a finding of concern and would normally lead to termination of drug development if this is discovered very early in development. On the other hand, we know some mode of actions which can lead to micronuclei which are not related to DNA damage. Secondary effects which are related to pharmacology, such as change of body temperature. Hypo and hyperthermy can lead in rodents to micronuclei, and this is not relevant for human clinical situation. With regard to the role of genotoxicity data in relation to carcinogenicity, this depends on the phase of drug development. In early development, when there are when no carcinogenicity data are available, the mutagenicity data are usually one factor to predict carcinogenic potential. And a positive genotoxicity, if it's a robust finding, may even result in a request to first assess the possible cancer risk before continuing clinical trials. That's a rare event, but in that situation, I mean, I'm not aware of such a request in Europe, but David, how do you say our friends on the other side of the Atlantic? I know cases where they have asked for a 6 months transgenic mouse bioassay in such a situation before continuing the clinical trials. And in the presence of carcinogenicity findings, the outcome of the genotoxicity studies are, of course, used then as part of the mode of action evaluation. If there is really indication that mutagenic mode of action is the key mode of action for tumor finding, then that would have dramatic influence on how to do the risk assessment. It's still the conservative perception that there would be no threshold, so you would not be able to define a real safe exposure range in this situation. Now this leads already to the second part of my presentation, carcinogenicity, and I will briefly discuss the current requirement and in particular the current problems with the current approach, and then talk about an ongoing ICH process with regard to a new proposal that hopefully will someday in the future will be an alternative to the rodent long-term studies. As for almost all small molecules, the carcinogenicity ICH guideline S1 is requesting to do 2 long-term studies with rodents. One is a 2-year rat study, and the other is either a 2-year mouse study or it is 6 to 9 months transgenic mouse study. And although these test models are the most expensive and also the most resource consuming, and in particular with regard to animal numbers, studies in the whole preclinical package, the results are very often of doubtful human relevance. That's a problem we have identified years ago. One of the more recent reviews which confirmed this finding has been published in 2011. In that review, 144 compounds which are authorized through European centralized procedure, they are on the market, have been reviewed for carcinogenicity testing. And out of these 144 compounds, 94 or 65% were positive in either mouse and or rat study. And in the vast majority of these cases, they are on the market, the findings were considered to be non-relevant for humans. Of course, this is a kind of unacceptable situation, and the need for a better alternative is quite obvious. And I'm absolutely glad that this point has been taken up by ICH three years ago. And I skipped this one, and I will now show you what the new proposal is, but in comparison to the current approach. So as I just said, the current approach is to start first with a 2-year rodent study. It's often positive, more than 50%. And of course, if you find tumors in rodents, you have to do a thorough assessment of these findings with regard to human relevance. And this assessment is usually a stepwise approach that you first look what is the mode of action for the animal tumors. Is this mode of action relevant to the human situation? And more specifically, is it relevant to the usually much lower exposure situation in clinic compared to the high dose scenario in the rodent carcinogenicity study? And this kind of human relevance assessment is usually done based on weight of evidence assessment on all the available data at the time point when such carcinogenicity study is completed. They based on the pharmacology, based on histopathology outcome from rat chronic study, based on genotoxicity, information about hormonal activity, immunosuppression, and so on. And now the idea for the new proposal is quite simple. Rather than having this weight of evidence assessment at the end of the whole process, the new proposal is starting with this weight of evidence approach. Starting in order to predict the outcome of the 2-year rat study and to predict the human cancer risk. And based on this prediction, we then ask, is there an added value to do the rodent carcinogenicity study? Do we need it anymore? Or is it already sufficient based on these weight of evidence assessment of all these knowledge to do the rat study? And this proposal has been, in fact, announced 3 years ago by an ICH regulatory notice document. In that document, this proposal is expressed as an hypothesis, and the hypothesis is that carcinogenicity assessment could be completed for certain pharmaceuticals without conducting the 2-year rat study, and that based on the knowledge of pharmacology and toxicology from other data sources, it should be possible to predict that pharmaceuticals will fall into 1 out of 3 categories. Category 1 is it's likely to be tumorigenic in humans. The product would be labeled as such, and no carcinogenicity would be needed. Category 2 would be there's uncertainty with regard to tumorigenic potential for humans, and there might be an added value to do the rat study. And category 3, this is split into A and B, is that it's likely to be tumorigenic in rats but not in humans. But the 2-year rat study would not add value. And 3B is it's not to be likely not to be tumorigenic in both rat and humans, and a 2-year study is not needed. So the regulatory notice document is continuing in saying that a carcinogenicity assessment document can be provided that would justify a waiver for the 2-year rat study. And obviously that would be the case for category 1 through 3A and 3B. Only in the case of a category 2, the 2-year study would add some value. And the carcinogenicity assessment document should take into account all this knowledge here, as I already mentioned earlier, pharmacology, genetic toxicology, and so on, including transgenic mouse study. In fact, there have been huge retrospective data analysis in order to show that this approach is feasible. One has been done by a group of pharmaceutical scientists which compared the carcinogenicity studies and the outcome of the rodent chronic studies, the rat chronic studies, genetic toxicology studies, and evidence for hormonal activity. And the conclusion here was that if all these 3 endpoints are negative, the likelihood that the carcinogenicity study is negative is very high. So you could predict a negative outcome. And we as European regulators have published a paper earlier this year where we have compared pharmacology of more than 250 compounds with the outcome of the rat study. And the conclusion here is that in many cases, the knowledge of pharmacology can be used to predict both the negative and positive outcome of the carcinogenicity studies. But although these are already quite convincing retrospective analysis, it was decided at the ICH level to do a prospective evaluation to evaluate this hypothesis. And this is an activity which is ongoing now for 3 years. So that sponsors are encouraged to to provide a drug CAD, making the prediction of the outcome before the outcome of the actual carcinogenicity study. I will skip the process, how this is evaluated here at the EMA. At the end, we are evaluating or have a consensus regulator's position on the predictive outcome, on the prediction of the carcinogenicity. The same would be for the actual outcome of the 2-year rat study, that it went through the same evaluation process. And in the end, of course, and that's the most critical part of this prospective evaluation, to compare what has been predicted with the actual outcome. And this will then give you an idea of the accuracy of this process. This template is a pivotal part of the CAD. I will skip this. Here is the current experience of the whole process. A status report was published at the ICH website. At that time point, it was last year in December, 25 CADs were evaluated. And what's interesting here is that there are clear differences in the categorical assignment between sponsors, industry sponsors, and drug regulatory authorities. In particular, with regard to category 3, 16 of the 25 were category 3 from sponsor side, and we accepted only, well, 6 plus 3, because the 3 were only partial alignment within agencies were also category 3. So, which means that the regulators have a more cautious approach and express more uncertainty and see more added value in some situation where industry is quite certain in their categories. Meanwhile, we have another 11 CADs and also meanwhile seven study reports of actual outcome. But I can't tell you what the consensus position of the regulatory authorities is. So important is, I think, the timeline. When can we expect a final decision on this process? The expert working group found it important, of course, to have sufficient category 3 cases, because only in category 3 there is a clear deviation from the current approach. And it was agreed that at least 20 category 3 cases, which means CADs and the actual outcome of the carcinogenicity study, are needed to make a final analysis and a final decision, and this is expected to be end of 2019. And based on the outcome of this analysis, then hopefully it's successful, I'm optimistic, then of course it will be discussed whether the ICH S1 guideline can be revised accordingly. So this, to sum up, of course, genotox carcinogenicity testing is a pivotal part of the preclinical testing for small molecules. The standard test approaches are well defined in ICH, but the data interpretation are not so easy. Yeah, I mean, that's of course a problem to make clear definition how to interpret. The key words here is, again, weight of evidence assessment and mode of action analysis, and normally this needs expert knowledge. And the relevant non-clinical findings can have a severe impact on overall benefit risk assessment, and usually cannot be overruled by clinical data, as expressed in one of my first slides. And of course, the very interesting ICH project is currently ongoing and hopefully will result in a revision of the ICH guideline. Thank you very much.